Hello everyone, and welcome to today's webinar titled Modeling Dissolved Oxygen. This webinar is sponsored by EPA's Water Quality Modeling Workgroup. My name is Jason Gilday. I'm with US EPA, and I will be moderating today's webinar, and thank you all for joining us today. First, I'm going to briefly summarize some of the features of today's webinar. We hope that you all have a lot of questions. If you do have questions, please type your question in the questions box on the control panel and click send. If your control panel is not showing, simply click on the small orange box with the white arrow to expand it. If you have any problems viewing the broadcast, just let us know in the questions box and we will post an answer in that same box. A phone number is also available on your control panel if you're having any trouble hearing audio through your computer speakers. This webinar will be archived for later viewing on EPA's Water Quality Modeling Workgroup page, and we will provide that archive link for you on a slide at the end of the webinar. Also, at the end of today's webinar, you will be able to obtain a certificate of attendance for participating, and we'll provide directions for downloading it at that time. The materials in this webinar have been reviewed by US EPA staff for technical accuracy. However, the views of the speakers are their own. They do not necessarily reflect those of US EPA. Mention of commercial enterprises, products, or publications does not mean that US EPA endorses them. So with those housekeeping items out of the way, let me get into the webinar series purpose. The purpose of this webinar series is to help water quality professionals better understand water quality modeling and how models can be used to solve the problems facing water quality regulators. Our target audience for this series is a water quality professional who may use, want to use, or review models as part of your job, but you do not necessarily have a modeling background. As such, we're assuming that the audience has a basic understanding of water quality science and concepts, but has limited direct modeling experience. Today's webinar is the 8th in our water quality modeling series. All of the past webinars have been recorded, archived, and posted to EPA's YouTube channel website. Links to the archived files are available on our Modeling Workgroup website. This URL will be shown at the end of today's webinar, and in a few weeks, a copy of today's webinar will also be posted on the same website. We encourage you to listen to any of the previous webinars or share access to those files with your coworkers. Today's webinar builds on the information presented in earlier webinars, and your feedback on the series and content has helped to shape each installment of the series. At the end of today's webinar, there will be a survey where you can provide feedback and additional comments. We encourage you to let us know how we're doing and what you like. And with that, I'm going to go to introduce our speaker. Our speaker today is Tim Wool. Tim is a national TMDL expert with the United States Environmental Protection Agency Region 4 office located in Atlanta, Georgia. Tim has over 25 years of experience in the development and application of water quality models that are used for TMDL development and waste load allocations. Tim established the Watershed and Water Quality Modeling Technical Support Center located at US EPA's Office of Research and Development Laboratory in Athens, Georgia. At the center, he was responsible for the development and establishment of the TMDL Modeling Toolbox, which brings together a collection of models and tools to streamline TMDL development. Tim is also one of the primary developers of US EPA's Water Quality Analysis Simulation Program, or WASP, which is routinely used in the US and throughout the world. Tim has supported US EPA with developing a technical approach for numeric nutrient criteria for the state of Florida, and he has significant experience in developing and reviewing TMDLs for bacteria, nutrients, metals, dissolved oxygen, and mercury. Before I hand off today's presentation to Tim, I'd like to make one final announcement. We will try to answer as many questions as possible throughout this webinar, and we'll be asking questions of our speaker at various points throughout the presentation. So please remember to type your questions into the questions box, and at I think it's 2 points throughout the webinar today, we'll pause and do a question and answer session with the questions that you type in. So with that, I'd like to hand things over to Tim to kick off today's webinar. Thanks, Tim.
Thanks, Jason. In today's webinar, we're going to focus on dissolved oxygen in the aquatic environment. We're going to look at some of the major water quality issues in regards to dissolved oxygen. We're going to look at dissolved oxygen water quality standards that are applied throughout the U.S. We're going to define the sources and sinks that affect dissolved oxygen, such as DO saturation, BOD, reaeration, productivity respiration, and sediment oxygen demand. Then we'll look at different levels of complexity in which we can model dissolved oxygen and present different models that could be used with examples of these different approaches. We'll begin by looking at some of the major issues in regards to dissolved oxygen in the aquatic environment. The first example we're going to look at is a dissolved oxygen sag curve in a riverine body. This is something that wastewater engineers have observed for over 80 years. In our example here, we have a point source coming into our river carrying oxygen-demanding materials as they're decomposed in the water. From that, we can look at the impact of this load on dissolved oxygen as we go through in distance. And typically, we get a curve like this, where at the very low point, we can find the critical concentration where we can look at the lowest level of dissolved oxygen in the water body, and we can adjust that the loadings to meet our water quality standards. One of the things that we have to understand here is everything to the left of the red vertical line, decomposition dominates and is occurring at a fairly rapid rate. Anything to the right of the red line shows that reaeration is more dominant and the water body is recovering. Right at the red line is where decomposition equals all sources of oxygen coming into the water body. In our example here, it's just reaeration. And then down towards the end is that recovery zone where we get into a more natural condition. We have recovery zone where plants and other things exist. Another example of impacts on DO are the creation of large hypoxic and anoxic zones in our estuarine and coastal waters. The amount of anoxic and hypoxic water has increased dramatically since the 1960s. In our case here, we're looking at the Gulf of Mexico, which is in the news all the time because of its hypoxic area. And these areas are created from large organic loadings coming down from the watershed, coming into areas where it can be gathered and accumulated and have large sediment oxygen demands from the bottom to decompose the organic materials. These areas of anoxic and hypoxic water is not just isolated to our estuarine and coastal waters. It has been observed in our lake systems as well. Again, it's caused pretty much by the same thing in organic enrichment, the accumulations of the materials, and oxygen consumed to decompose those materials. Next, we'll look at some samples of water quality standards that are used for dissolved oxygen through the United States. It's these standards that would kind of dictate what kind of modeling approach we would use to either develop a waste load allocation or a TMDL to be protective of DO. All right, let's look at some of the more prevalent dissolved oxygen water quality standards used in the United States. The most widely used one is a never to exceed value. In other words, DO will never be less than 4 or 5 milligrams per liter. Or a little bit more of a hybrid onto that one is never to exceed those values more than X% of the time. So typically it's like you can exceed it but no more than 10% of the time. Another one is no less than 4, but you must maintain a daily average of 5 milligrams per liter. And then one that's becoming more prevalent now is actually developing a water quality standard for dissolved oxygen that maintains a certain level of oxygen saturation in the water column, and that would be maintained throughout the night cycle. Another method for dissolved oxygen is one of resource needs. In other words, it looks at sensitive species, spawning periods, periods where juveniles are in certain portions of an estuary where DO requirements are needed. So it allows to have a multi-tiered DO approach that will break out surface waters from bottom waters to protect invertebrates and such in the bottom. So it's basically a site-specific DO criterion based upon sensitive species in a particular water body. So we can see that dissolved oxygen standards are varying quite a bit, and you can see how models would be very, very useful for determining how to set load allocations and waste load allocations to meet the desired standards. Let's take a moment and refresh our memories on the different sources and sinks of dissolved oxygen in water, and also look at some of the other things that can affect how much oxygen can be held in the water. All right, let's look at the dissolved oxygen cycle. One of the primary sources of oxygen to the water column is reaeration, which is the exchange of oxygen from the atmosphere into the underlying water column. And there also could be an exchange from the water column back up to the atmosphere depending upon the concentration of dissolved oxygen in the water. A sink for dissolved oxygen from the water is through biochemical oxygen demand, and this is basically the consumption of oxygen as bacteria go through a metabolic process to break down these materials. And then you have sources and sinks from algal or biotic components, whether it's algae, benthic algae, or macrophytes. You have productivity, which will increase or add oxygen to the water column. And then you have respiration, which will remove oxygen from the water column. And then another source is sediment oxygen demand, and this is the biochemical, geochemical processes that go on in the underlying sediments where carbonaceous and organic materials are broken down, and it exerts a demand on the overlying water column for oxygen, and in certain instances causes a nutrient flux from the sediments back up into the water column. All right, our next steps are we're going to look at the different things that are sources and sinks for dissolved oxygen in the water column, and we're also going to look at other phenomena that occur in the water column that affect the dissolved oxygen concentrations. The first one we're going to start off with is saturation, which is the amount of oxygen that can be held by the water. The amount of oxygen that can be dissolved and carried within the water column is controlled by several factors. The first and probably the largest factor is the temperature of the water. So you can see from this graph, the colder the water is, the more oxygen that can be dissolved. As the water gets warmer, the carrying capacity of dissolved oxygen goes down substantially. The other thing that affects the amount of oxygen that can be held in the water is the salinity or chlorinity concentration in the water as well. The higher those values, the less oxygen that can be carried. Another factor that controls how much oxygen can be dissolved in the water column is the elevation that the water body is at. The higher the elevation, the less oxygen can be dissolved and carried throughout the water column. So you can see here that at sea level, the dissolved oxygen saturation points are much higher than, say, at 3 miles. The table that's presented here is from the Rates, Constants, and Kinetics Manual that was published in 1985. This table just shows you the dissolved oxygen saturation concentration at different temperatures, assuming 1 atmosphere of pressure and no salinity. This is a handy little reference just to look up to find out what the saturation point is for a given water body at a temperature. We're going to look at another phenomenon that occurs in lakes, estuaries, and deep rivers, and that is stratification. Okay, let's look at stratification in estuaries, lakes, and deep rivers. When a water body is completely mixed from top to bottom, no matter what the depth is, we expect to see a profile much like the one given here, where the dissolved oxygen concentrations in the surface water and the bottom waters are approximately the same. The bend of this curve is actually really a function of how deep the water body is. What stratification is, it's a buildup of a density gradient between the surface and bottom waters. This gradient is typically caused by temperature, salinity, or both. Once stratification occurs, the surface waters are disconnected from the bottom waters, and the transport into the bottom, or the mixing with the surface and the bottom layers, is greatly diminished. While there is still mixing across that gradient, it is way more minimal than if it's not stratified. So that basically disconnects the surface waters from the bottom waters. So the surface waters are mixing, and getting the benefit of reaeration and productivity. So dissolved oxygen would be much like it is in a river. And the bottom waters are disconnected and are only mixing amongst themselves and are being subjected to sediment oxygen demand and minimal transport of dissolved oxygen from the surface waters to the bottom, which leads to profiles that look much like the graph here, where we have pretty much constant concentrations of temperature and dissolved oxygen in the overlying water column and depleted oxygen and cooler water in the bottom. The next component of the dissolved oxygen cycle that we're going to look at is carbonaceous biochemical oxygen demand. First thing we have to understand is, what is BOD? And BOD is something that we do not measure directly, but measure it indirectly. So the measurement is actually the amount of oxygen consumed by bacteria during the decomposition of organic materials. The organic materials can come from many, many different sources. Some of the more prominent ones are wastewater treatment plants, organic materials washed off from watersheds, organic materials from decaying biotic components in the water body itself, such as algae, macrophytes, periphyton. The typical measurement for BOD is what's called a 5-day BOD. And what this represents actually is the amount of oxygen consumed during a 5-day period and measured and expressed in oxygen equivalents. So it's pretty much the quantity of the decomposed organic carbon expressed in equivalent oxygen units after 5 days. Another method for measuring carbonaceous biochemical oxygen demand is what's called a long-term BOD. What the long-term BOD measurement does is it calculates the amount of oxygen that would be used to completely deplete the organic material. And it's typically done over a 120 day period of time. The issue with this is it's very labor and time intensive. So if we look at a typical long-term curve, we would have the amount of oxygen that's consumed over this 120 day period and calculated the oxygen equivalents of the CBOD. Our measurement from the 5-day would give us a value here. But if we play it out for the 120 days, the long-term BOD rate is substantially higher. So we call this ultimate BOD. It's the amount of oxygen consumed to totally deplete all of the organic material. So the 5-day rate just gives us the amount of oxygen consumed in a 5-day period. The long-term one gives us the total oxygen consumed to deplete the organic material, or the ultimate BOD. The Georgia Environmental Protection Division has developed a piece of software that will take the laboratory measurements for the long-term BOD test, and you would put it into this program, and it would do some separations for you, calculating some various components that are very useful in modeling. The benefits are we can characterize different sources of BOD because they aren't all the same. That's why a lot of our water quality models have multiple state variables for BOD so that we can characterize each source individually. It'll calculate the BOD decay rate that you would put into the model. It would calculate a nitrification rate that goes into the model. That would be the amount of oxygen consumed by the conversion of ammonia to nitrate. And then lastly, it'll give you an F ratio for converting a 5-day measurement to a long-term measurement, which is very handy, which means you don't have to do long-term measurements over long periods of time for each individual samples. This slide here gives you magnitudes of decay rates and concentrations of BOD in untreated sewage. So the bottled KD rates are actually the BOD decay rate. So for untreated waste, it's typically 0.35 per day, primary treated 0.2, and then activated sludge 0.05. And these rates have changed dramatically over the last 20 to 30 years as our treatment processes become much more advanced. Just to give you an idea of concentrations for untreated sewage in developed countries, it's somewhere between 200 and 250 milligrams per liter, and a little bit higher for developing countries. The next process that we're going to look at that is a source to dissolved oxygen is reaeration. Reaeration is the transfer of oxygen from the atmosphere to the water column. It's controlled by water depth and velocity, which we call the hydraulic reaeration rate. The shallower the water and the faster the water velocity, the more oxygen is brought into the water column from the atmosphere. Other things that affect reaeration are wind speed, the wind blowing across the water surface, waves and turbulences, stream ripples, and water control structures such as dams. Reaeration is not always oxygen coming in from the atmosphere to the water column. You could have a transfer of oxygen from the water column to the atmosphere if the oxygen concentration in the underlying water column is greater than its saturation point or the amount of oxygen it can hold. At that point, the oxygen will leave the water column to the atmosphere using the same rate that would be calculated for bringing oxygen into the water column. There exist several methods for measuring reaeration. They are time-consuming and expensive and are typically done at a single condition, which would be the period of time that the analysis is being conducted in the water body. If the reaeration rate is being measured at the critical condition, it's very, very useful because it's one less unknown that you would have to put in the model. If it's not at the critical condition, it's going to have to be extrapolated to the condition that you're modeling to. Now that we know what reaeration is, we can look to see how reaeration is calculated in models. This slide shows three of the more predominant reaeration equations that are used for rivers and streams. These equations were generated by looking at field data and regressing out and coming up with this empirical relationship of reaeration as a function of water velocity and depth. Because those reaeration equations were generated from field data to come up with the empirical representation or fit to that field data, they are better suited for the ranges of velocities and depths in which there were actual measurements. So this table here, which was developed by a gentleman by the name of Kovar, tells you that if in a specific velocity and depth regime, which one of those reaeration equations are better suited for calculating reaeration. Another source of reaeration or loading of oxygen into a water body is water falling over a control structure such as a dam. And again, here is another empirical relationship of the oxygen load coming into the downstream water as a function of water falling over a dam. This relationship is taken from Steve Schaffer's 1997 surface water quality modeling book. And it takes into account the height of the dam, the difference in water quality above and below the dam, and the water temperature. Another major component of reaeration is wind-driven reaeration, which is important for estuaries, lakes, and reservoirs, and any water body that has a large surface area. It typically can be ignored for rivers and streams. The wind-driven reaeration function that we're looking at here was one developed by Donald O'Connor in 1983. And the major forcing function is going to be the wind speed, the air temperature of the atmosphere, and then the water temperature of the water column. And it'll predict or calculate the reaeration rate as a function of the wind speed. We call this the wind-driven reaeration rate. The next process in the dissolved oxygen cycle that we'll have a quick look at is productivity and respiration. Productivity is going to be defined as the amount of oxygen that is produced through photosynthesis for phytoplankton, periphyton, macroalgae, and macrophytes through the daylight period. Respiration will be the amount of oxygen that is consumed due to metabolic processes within the phytoplankton groups. So if we look at a typical daylight cycle, during the daylight period of time, the phytoplankton are undergoing photosynthesis and they're producing dissolved oxygen. Throughout the whole day period, the phytoplankton are also depleting oxygen as part of their normal metabolic processes. If you are simulating dissolved oxygen in a water quality model where there's high productivity, it's going to be very, very critical to take into account both the day and night cycles of the phytoplankton and its impacts on dissolved oxygen. The last process that we're going to discuss in the dissolved oxygen cycle is sediment oxygen demand. Sediment oxygen demand and subsequently nutrient releases is a function of sediment diagenesis or decomposition of oxygen-demanding materials in the underlying sediments. These sediments are recipients of the settling of dead biotic material, organic materials from the watersheds, and are actually a function of the external loadings to the water column. Sediment oxygen demand is a major sink for oxygen in the aquatic environment and is responsible to leading to large areas of hypoxia and anoxia in estuarine and coastal waters. Sediment oxygen demand in coastal and estuarine waters has led to harmful algal blooms, jubilees, which is periodic anoxic hypoxic conditions in the underlying waters, which cause large fish kills and things like crabs and such to leave the water body up onto the shores, flora and fauna death, and death to benthic populations. Sediment oxygen demand cannot be ignored. It needs to be included in our models when we're modeling the dissolved oxygen cycle. EPA's Region 4 Field Services Group, who is one of the primary developers of the in-situ SOD measurement, has measured SOD in water bodies in Region 4 and throughout the country. It's never failed to measure an SOD rate. A typical natural SOD rate is approximately 0.08 grams of oxygen per meter squared per day in the water body. This table here just shows you the range and variability of SOD in some rivers measured in Region 4 states. So how do we typically determine SOD? Sometimes we have sporadic measurements that we could apply to the water body that we're interested in. It could be a stream close by or a reference stream. But more likely than not, we calibrate it into the model with an unknown. And in our example here, we showed the blue line up here, which is a model prediction without including SOD. And then we have our measured values of dissolved oxygen below. And we actually just calibrate in and change our SOD rates until we match the observed data. While this looks pretty good, it's probably a bad idea because we failed to link our external sources to a response in the water body. One way around this is that we can actually go out there and measure it. There's a couple of different protocols available for measuring SOD. Both are time-consuming and they are expensive. One would be the placement of an in-situ chamber on the bottom of the water, and it measures the amount of oxygen consumed through a time period. Or you could take cores and bring them back to a laboratory and do the same thing. That's how we can measure it. Unfortunately, that has no way of relating the SOD to the external loads. So it's kind of critical because sediment diagenesis is driven by the organic fluxes from the water column, and which are ultimately driven by external loadings to the system. So what we're missing here is the missing link is we know that there's SOD out there. We can measure it, but we don't know exactly what caused it and how much came in. So we need to come up with a way to link the cause to the effect. There have been many TMDLs and waste load allocations developed in the United States that did have an SOD component. Unfortunately, when the load reductions were calculated, there was no real good way to attenuate the change in SOD as a change in the loads. And in essence, we may have been over-regulating or not accounting for the changes in sediment oxygen demand as a function of the overall loadings to the system. So how can we account for changes in sediment oxygen demand as a function of regulatory or load reduction activities? Well, there are several water quality models out there now that include the ability to predict changes in sediment oxygen demand and nutrient fluxes as a change in external loadings. Qual2K, CQual-ICM, CQual-W2, and WASP are a list of some of them. And it's very, very useful as we go through the TMDL process and the waste load allocation process that we can predict sediment oxygen demand and nutrient fluxes under the current condition, but also can predict the sediment oxygen demand and nutrient fluxes under the TMDL or the waste load allocation system. This seems to be a fairer way of doing load reductions. All right, we're going to pause in the webinar right now to take any questions in regards to the dissolved oxygen cycle in the aquatic environment.
Great, thanks, Tim. And we've got a few questions that have come in so far. I'll get to those in a second. Maybe I'll just pause here and remind folks to, as we're going along through the presentation, if you think of something, just type it in the question box, and we'll get to it during these question and answer sessions. Also, we'll give you a few, give you another minute or two here to be typing your questions in for Tim. I guess maybe a couple more reminders that, this will be archived and posted to our EPA TMDL modeling website. So if you want to go back and revisit things, that'll be up there on the YouTube website. And there will be an email going out after this with the link to various websites, a link to a certificate for participating today and such. So with that, why don't I just get into the questions? I see some more popping up, which is great. So Tim, I guess first of all, maybe I'll comment real quickly that we're focusing today largely on the technical aspects of dissolved oxygen modeling. I know there are a lot of kind of more policy and standards related questions that are out there, and I see a few of those in the questions that have come up. For those, I would generally ask folks to contact your local state or EPA representative, whether that's somebody at the state or within an EPA regional office. There's also a number of EPA websites that talk about kind of dissolved oxygen standards and policy. So you could easily find that kind of going into Google and typing those search words in. And in the webinars we do here, therefore, we'll avoid more of those policy-related issues and leave those up to the people who are experts in those areas. So with that, I think the first question, Tim, would be, what do you do when you have a hypolimnetic lake, or where you end up with anoxia, hypoxia in the deep area, and how do, in general, that you've seen, do standards apply to those waters versus like upper layer of waters?
Yeah, I saw that. That's a great question, and I think I'll refer to the Virginia province method for developing a site-specific dissolved oxygen standard. Takes into account the different portions of the water column and what's living in the water column, and comes up with dissolved oxygen concentrations and frequencies that will support recruitment of and healthy aquatic organisms. In reservoirs and such, it's a little bit more difficult because we have reservoirs here in the southeast that are mostly anoxic most of the year. Most of the fish and such in those types of systems stay up in the upper waters. But coming up with standards for stratified systems definitely would using a method something like the Virginia province method, where it's resource-based.
Thanks, Tim. Question number two, when modeling DO depletion, do you attempt to incorporate estimates of BOD that can arise from a result of, say, a cultural eutrophication, say a phytoplankton biomass that results from nutrient loads, which could be a substantial component, particularly for lakes and reservoirs?
Yes, I mean, that's one of the reasons why a lot of the water quality models have now added multiple BOD groups. The typical practice now that we use here in Region 4 is we'll have at least 3 different BOD groups. One would be representative of the watershed BOD, organic loading coming down. The other would be of wastewater treatment plants. And the third would actually be components of the impacts of phytoplankton, periphyton, and macrophytes on BOD as they die. So yes, we do include it.
Great. We have a question from someone doing some groundwater work. Looks like they have maybe some low dissolved oxygen levels coming in from some influent groundwater, which is below most permit limits. The question is, what are the typical sources and sinks for such low levels in groundwater?
I mean, the biggest problem with dissolved oxygen in the ground is there is no way possible to get reaeration down there, so it's going to be a function of it just being disconnected from the atmosphere. There is no productivity. The DO levels are going to be low.
Yep. Can you expand on the consequences of DO depletion, specifically the mobilization of certain metalloids and metals such as arsenic and manganese?
Yeah, I thought about adding a slide on that. There's not a whole host of models that have the capability of including the impacts of the changes in dissolved oxygen concentration and the mobility of metals. One of the models that come to mind that takes into account that is Sequal W2. It takes into account, I believe, iron, manganese, and a couple of others down in the hypolimnium of the dam pools for changes in its structures and also depleting oxygen.
Thanks, Tim. A question about models in SOD, and maybe you're going to get into this. The question is, how do models predict changes in SOD?
Yeah, we won't talk about that too much in the models, other than I'm just going to show you how important SOD is. But the way there's two ways to do it. One is you can use descriptive sediment oxygen demands under your current condition. And there are some equations that you can estimate what the SOD would be once you reduce the productivity in the system and reductions in the carbon load. The way we're doing it now is the water quality models have the sediment diagenesis models hooked up to them. So we use the current condition to get the models calibrated and compared to whatever data we have in the water column, in the sediments in the water body. And then once we do load reductions, we would rerun the sediment diagenesis model, and it would calculate the change in sediment oxygen demand, but also the change in the nutrient fluxes from the sediments back to the water column.
And does the significance of sediment oxygen demand vary by water body type?
Yes, it does vary. It not just varies by water body type, but it varies pretty dramatically within a water body itself. If you have areas of high deposition or pooling or in reservoirs where you have a lot of settling, the rates tend to be higher. But they do vary dramatically across water bodies.
So lots of SOD questions, which is good. Maybe I'll lump these all together here. Since SOD takes a long time to get established, what is the time period that sediment diagenesis model is run to establish SOD for a scenario?
Well, it depends upon the system that you're in, and working with some of my colleagues, Jim Fitzpatrick at Hydroqual, sometimes we spin the model up for about rerunning a series of time series for about 100 years to bring the model up into equilibrium. For some other systems, I've seen it come into equilibrium much faster than that. We've also made some improvements and changes to the sediment diagenesis model so we can reset or set some of the initial conditions so that it gets to the equilibrium faster. But it does take time.
And then another SOD question. If you have, say, a large historical organic load already present in your water body, how quickly would it take SOD to decrease as current loads in the systems decrease?
Yeah, so I mean, there's no real book answer to that because it really depends upon the magnitude of the previous load. It also depends upon if there's scouring or any other transport phenomena that are going on in there. But responses are, if it's a quiescent system like a reservoir or a lake, the response times will be fairly long, 40, 50 years.
And I think the last SOD question so far, you talked about some regional SOD rates for Region 4 that you guys have been working on. Are there other similar things that have gone on in other regions? Say the question is specifically for Region 6, but maybe even others within the U.S.?
I'm pretty certain that there are some state programs that have adopted and use the SOD chamber protocol as well as the core protocol. I know that our field services group have actually done SOD measurements throughout the United States and actually outside the United States for that matter. I don't know specifically, but I do know that a lot of states, where there's a large water quality issue, they will invest the time to either measure the SOD themselves or contract it out.
Great, thanks. A couple of questions on WASP-8, which you mentioned, so maybe I'll let you do a little advertising. When is that scheduled to be released and downloaded, available for download?
Well, it was scheduled to be released on Friday, but somehow I got talked into doing a webinar. But
which we thank you.
Yeah. So my goal is we're running it through our QA/QC process right now, and actually one of them failed, and we got to figure out why. I'm hoping to have it released by Friday.
Wow, great. Maybe I'll chime in with one quick thing. There was a question on EPA regions. So for those of you that don't know, the U.S. EPA is separated into 10 regions across the U.S. The regions are tasked with covering a variety of states within that region. And so when we talk about EPA Region 4 versus 6, that's a matter of those two specifically are the southeast and kind of south central are those two. And if you could look up EPA regions on Google for anyone who has any more questions about that. And then maybe we should probably get going again, but one more question maybe. Will the 1985 Rates, Constants, and Kinetics book that you mentioned be updated, and if so, do you know when?
Thanks to the efforts of Region 6 and Region 10, there's an effort underway to update it right now. There are drafts of the new version being shared, so hopefully not too far out.
Great. I have a lot of extra questions here. I'm going to suggest that we may perhaps move on to part 2, and we're going to have another question and answer session. I'll just kind of keep prioritizing questions as they come in, so keep typing them in even while Tim's talking. And we'll try and get to as many as we can once the webinar is done. And thanks, everyone, for the interest in the questions. Tim, back to you.
Okay. Okay, we're going to focus our attention now on modeling dissolved oxygen. We're going to look at potential water quality standards and the modeling approach that should be used to investigate meeting that standard. We'll talk about modeling assumptions and what it means in decisions that are made using modeling tools. Okay, we're going to go back and revisit some of the water quality standards that were described at the beginning of this webinar and talk about what is the appropriate modeling approach to investigate meeting those standards. The first one is never to be less than 4 or 5 milligrams per liter at any time, anywhere. This pretty much allows us to use a steady state critical condition, because the assumption here is if you can meet the standard at the most critical condition, you'll meet the standard everywhere and all the time. You can also use time variable models in place of steady states. It's just that the solution technique is different and may have to iterate to get to that point. Never less than 4 milligrams per liter, daily average of 5. You can again use the steady state model with the critical condition, but you also have to consider the diurnal effects, change in water temperature, productivity, respiration, or you can use a time variable model. Never to exceed a concentration 4 or 5 milligrams per liter more than X% of the time. This would require the use of a time variable model running through a continuous simulation, which will take you through all the various critical, subcritical conditions within the period of time that's being investigated. Percent dissolved oxygen saturation, you could do this either with steady state with a diurnal option or time variable. So you're looking to determine what is the maximum loading that you can have but still achieve a percent saturation. The Virginia province or the resource-based standards pretty much going to require a time variable and a multi-dimensional model to look at load reductions that could be implemented that would protect various resources at different points of the water body at different times. Okay, now we'll just go through a list of potential water quality models that are capable of simulating dissolved oxygen in the aquatic environment. This isn't a full comprehensive list, but this is a list of probably the most popular approaches and models that are used in the United States to date. First, we need to acknowledge that there are many spreadsheet calculators out there that states have used for developing waste load allocation for carbonaceous materials and to meet dissolved oxygen endpoints. They usually employ a simple Streeter-Phelps calculation where they're calculating the impacts of carbonaceous materials on dissolved oxygen. They do not take into account diel variations. They are not predicting temperature. It's a single temperature. They may or may not have SOD, and they don't include productivity and respiration. So it's a very, very simple approach. Along with spreadsheet tools, there are many, many dissolved oxygen SAG models that are used throughout the United States. The one I'm going to use as an example here is the Georgia dissolved oxygen SAG model. It's a one-dimensional model. You can download it from the link that is shown here. And it employs a modified Streeter-Phelps. It is steady state. It does not calculate diel variation. It includes a single CBOD group. SOD can be inputted and as a descriptive SOD rate. It is not simulated. You can input a net productivity, which is basically production minus respiration, to account for biotic production of dissolved oxygen. This is a very good tool because it is relatively easy to set up. It has a nice interface, good documentation, and has been well thought out. The next potential water quality model is Qual2K, and it's probably the most widely used steady-state water quality model used in the United States and throughout the world. It does steady-state calculation. It does allow a diel variation. It simulates temperature and the diel changes in water temperature. It has the ability to do multiple BOD groups. This way you can either look at different discharges a different way or account for different reaction rates for BOD. It can do production and respiration of DO either through algae and or periphyton. You can describe SOD to the model, or it can predict the SOD as a function of the external loadings to the model. Again, a very, very, very capable model. The first one-dimensional time variable model that would be very applicable to doing DO analysis in the aquatic environment is EPD RIB1. It is a one-dimensional hydrodynamic and water quality model. It has the ability to model diel variations. It simulates temperature. It can do a single BOD group. It also has a component for doing NBOD if needed. It can simulate production and respiration of dissolved oxygen from algae, and you can also describe SOD to the model. It's a very good dynamic model, and it's available from the website listed. The next potential water quality model is CQualW2. It's probably the most widely used water quality model for reservoirs in the United States. It can be applied in one or two dimensions. It's a dynamic model capable of predicting diel variations in temperature and DO. It simulates temperature, has the capacity to model multiple BOD groups, can include the impacts of production and respiration from multiple algal, periphyte, and macrophyte groups, that describe the SOD rate to the model or have it simulated as a function of a sediment diagenesis model. The model can be downloaded from the website listed. The next water quality model is EFDC. It's an integrated hydrodynamic and water quality model that can be applied in 1, 2, or 3 dimensions. It includes diel variations of temperature and dissolved oxygen. It simulates temperature. It can do multiple BOD groups, include the impacts of respiration and production of dissolved oxygen from algae, periphyte, and macrophytes. The user can either describe a sediment oxygen demand or could be simulated using a sediment diagenesis model. All right, the last potential water quality model is WASP, which is a 1, 2, or 3-dimensional model. It is a dynamic model that can include diel variations. It simulates temperature, can handle multiple BOD groups, has multiple algal, periphyton, and macrophyte groups. You can either describe SOD to the model or have it simulated using a sediment diagenesis algorithm. Okay, next we're going to look at a couple of different approaches for simulating DO with water quality models, starting from a very simplistic approach through including the impacts of production and respiration. For the scenario modeling, we're going to be using our dissolved oxygen doseg example from the beginning of the webinar, where we have a load coming into our river system, and then we have an area just downstream of the load where we're not meeting standards. We're going to be setting up a steady state model to look at the load reductions to meet standards. What we're doing is we're going to simulate a critical condition, which is July 4th, 2012. It had the lowest dissolved oxygen measurements with the highest water temperature, and we'll be using a 7Q10 flow, which is the lowest 7-day average flow in a 10-year return period. Water quality standards we're going to meet are twofold. We'll not allow the DO to go below 4 milligrams per liter and maintain a daily average of 5 milligrams per liter. The modeling approach will be steady state with the diel variation in temperature and solar radiation. Our first modeling scenario that we're going to be using is the Streeter-Phelps with that SOD. This is as simple as you can get for modeling dissolved oxygen in the water. We'll include the impacts of reaeration and oxygen consumed by carbonaceous biochemical oxygen demand. We set up our model, and we are going to take into account diel variations in temperature. You can see that there is about a 3-degree temperature change in our water body during the summer. We'll also be looking at diel variations in dissolved oxygen. It changes slightly through the day-night cycle. And we'll also be calculating from the predicted dissolved oxygen concentration an average daily DO concentration. So we'll set up the model and we run it under the current conditions. And we'll be seeing a graph like this over and over in the webinar. The blue line is the predicted DO concentration through the 24-hour period. The green line is the daily average predicted dissolved oxygen concentration. The red line is the minimum DO criteria, in other words, the 4 milligrams per liter. And the 5 is the daily average of 5 milligrams per liter. So we would do our reductions to make sure that both the blue and the green line fall. The blue line falls above the red line, and the green line falls either on the black line or above the black line. So when we run our model, this is the fluxes that were calculated in grams per cubic meter per day. And we can see that in our case here, we just have a flux for BOD and reaeration. We would go ahead and make our reductions with the model until the blue line is above the red line and the daily average concentration is equal to 5. So using the simplistic approach to meet these water quality criterion, we needed a 40% reduction in BOD. The next scenario we're going to look at is a modified Streeter-Phelps. We're going to include the impacts of sediment and oxygen demand. This represents probably the most simplistic setup you should use when investigating impacts on dissolved oxygen. We'll be doing pretty much everything the same way we did before, but in our case with this model scenario, we've added SOD to the model, and you'll see that our predicted dissolved oxygen concentration and daily average oxygen concentrations have been reduced. We now have a new flux introduced to the model, and sediment oxygen demand makes up a 6% loss of oxygen from the fluxes in the model, which when we go to do our reductions, we now have a greater reduction in BOD to meet the dissolved oxygen concentration. It went from a 40% reduction to a 53% reduction in BOD. The next scenario, as we keep building on these scenarios, is the Streeter-Phelps with SOD, plus we're going to have the impacts of nitrification, the conversion of ammonia to nitrate on the dissolved oxygen concentrations. And again, much like before, we have our setup. Now that we've included nitrification as well, you'll notice that our dissolved oxygen concentrations and daily average concentrations have been reduced even further. And now when we look at the fluxes, we can see that we have a new flux in there for nitrification, and 10% of the oxygen flux in our model network is lost to nitrification. When we go and do our reductions now, not only can we reduce our BOD, but we also can reduce ammonia in our system. So to meet our water quality standards, we needed a 64% reduction in BOD and a 38% reduction in ammonia. These numbers can be changed slightly because you can trade off on the impacts of dissolved oxygen between the two components. The last scenario we're going to investigate using our river model example is a full DO balance, where we're going to include the impacts of phytoplankton's photosynthesis and respiration on the dissolved oxygen cycle. As like before, we set up our model and we include the growth of phytoplankton, and we get a curve under our current condition that looks like this. You'll notice in this case here that actually the instantaneous dissolved oxygen concentration during the daylight periods actually is bumped up higher than on our previous simulations. So we definitely can see the impacts of the productivity in our initial model runs. When we look at the fluxes now, that we can see that productivity is adding 2% of the oxygen to the system, and respiration is just a little less than 1%. As we go forward and we apply our reductions, we'll see that including phytoplankton, our reductions in BOD went from 63% to 56%, and our ammonia reductions went from 38% to 50% to meet the water quality standard. In this case here, the most critical water quality standard was the daily average 5 milligrams per liter. Clearly, it made the 4 milligram per liter instantaneous value quite easily. This next section is going to give you a couple of examples of using the time variable water quality models to predict dissolved oxygen, so that we could take into account not to exceed X% of the time of a standard, look at percent dissolved oxygen saturation and exceedance values for it, as well as how it can be used to calculate hypolimnion and oxic-hypoxic volumes. Our first set of examples use steady-state models to look at different scenarios in which you can model dissolved oxygen. We're going to look at a couple of examples where we're going to use the time variable models to look at impacts on dissolved oxygen. It should be clear that you can use the time variable models to make the same calculations as the steady-state models. They're all capable of doing that. The only difference between a steady-state and a time variable model is a steady-state model assumes everything's in equilibrium, there's no changes, so it can use a more simplistic solution technique versus a time variable model where it has to iterate to get to the steady-state answer. The time variable models can be used to calculate a percent exceedance of a criteria, whether it's dissolved oxygen concentration, percent dissolved oxygen saturation, calculate a hypoxic and oxic volumes, and when it also can account for the effects of stratification and destratification. Okay, we're going to look at another way to look at model output for allowing a 10% exceedance of an instantaneous value. In our case here, we have a lake example where we have 2 complete years of simulation. We have the 4 milligram per liter DO standard. But in the case here, the standard allows for a 10% exceedance. So if we take our current example here prior to doing load reductions and we do a probability distribution, we'll see that it exceeds the 4 milligrams per liter of about 58% of the time. So ultimately, when we do our load reductions here, we would have to reduce the model loadings until the DO line became above the 4 milligram per liter at the 10th percentile. Another dissolved oxygen standard that's becoming a little popular with the states is expressing the DO criterion in a percent saturation. This allows for states that have warmer waters to develop more realistic DO standards. Works pretty much the same thing as the instantaneous values, but instead of being a concentration, a fixed concentration of like 4 milligrams per liter, it is a percent saturation of dissolved oxygen in the water column. And we'll remember from earlier in the presentation, that percent saturation, or the amount of oxygen that can be carried in the water, changes as a function of water temperature, elevation, and salinity. So in our case here, we have our lake, and instead of looking at the instantaneous 4 milligrams per liter, we're looking at the calculated percent saturation in our lake. And you'll notice that there's periods of time that the percent saturation actually is above 100. That's because in our lake there's large algal blooms that occur, which it's making the DO concentrations go above the saturation point. But we would still do the same thing when it came to developing our load reductions. We would develop a scenario that would reduce our loadings so that our dissolved saturation point would go above the 64% at the 10th percentile. So that's just another way of looking at another endpoint. Okay, another way to look at model output is actually to animate your model in space and in time. And this, we're looking at the bottom layer of our lake model, and we want to just look at areas where we have. I'm not sure what happened there, but just the point here in the lake model is that you can both in space and time look at the predicted dissolved oxygen concentrations, and you can actually calculate volumes of water that are below a resource. This completes a resource need for aquatic life. So with that, we're at the end to take questions.
Great, thanks, Tim. And I see a lot more questions here, so give me a minute to transition slides over, and we'll get going with those. Again, keep typing those in. We do have quite a bit of time left, so that's the end of the presentation part of the webinar, but we'll take as much time as we need here to answer the questions we receive, and then we'll push out a few more slides talking about how to take the survey at the end and how to get the links and certificate. So with that, ready for questions, Tim?
Sure.
So first question is kind of related to I guess, steady state models and the fact that photosynthesis and respiration are cyclical. So can you maybe explain, give a little more explanation how a steady state model works and how it takes into account those daily fluctuations?
Yeah, so when we say daily fluctuation, we're just talking about a diel variation. And the diel variation is actually a function of making the steady state calculation like on an hourly time step, where things like solar radiation, air temperature, dew point are updated, and temperatures are recalculated. Phytoplankton productivity is affected by how quickly they're growing at that time based upon their light needs. So that's how it kind of works in a steady state situation. It's hard to determine whether or not in a critical, most critical condition that is the most critical condition to be protective against productivity and respiration. The way it works in time variable models, if you actually parameterize the model correctly and you believe that period of time that you use in the steady state model is a critical condition, as the time variable model approaches that, it should show it as the critical condition as well.
Great, thanks, Tim. And got a lot of questions about sediment and SOD and other things. I'm actually thinking perhaps we should get you guys to do a sediment and SOD webinar one of these days. But with that, a couple more questions on that topic. How responsive is sediment diagenesis to the organic loading rate?
When you say the organic loading rate, so there's two organic loading rates. There's the organic loadings that are coming down from the watershed into our receiving water body, and then there's the settling out of the mixture of those organic rates and the biological or biotic settling rates in the water column. It is very, very sensitive to that. Just imagine, you know, during periods of time where you have high productivity, you have phytoplankton that are naturally dying and are settling out to the bottom. That is direct fuel into that system. So they do respond fairly quickly, but also think of the sediments as like a battery that gets charged up from all of these loadings through time, and it discharges at a slower rate than what it's loaded as.
Great, thank you. Another question kind of on organic loading. In the models you've talked about kind of the existing organic load, but how would you go about determining if your load is a natural load or an anthropogenic load in your stream?
So typically how we do that is most of the modeling we do is watershed based. So we're using watershed models linked to the receiving water body models. And we can determine the difference between anthropogenic and non-anthropogenic by looking at the land uses associated with it as well as the direct discharges. So we can separate out and know the difference where each component is coming from.
Great, thanks, Tim. A question on models in general and ice cover. Can you talk about how ice cover in general affects DO and how models might go about accounting for that?
Yeah, so like W2 and WASP have the ability in their temperature models to generate ice. And ice, once ice is formed, of course, you're disconnecting the atmosphere from the underlying water column. At thinner layers of ice, there is still a transfer of oxygen to the water, but once ice is full on, the reaeration rate is cut off, as well as the light is degraded going through the ice into the water column. So it has a big effect.
Yep. A question about kind of metals and chlorides specifically. Do those kind of concentrations affect DO in a stream or lake?
Well, chlorides definitely affect the saturation point. Metals will affect the available oxygen because some of the metals, like iron and such, will remove oxygen, dissolved oxygen, from the water column to adhere to the iron. So they do have an effect for sure.
Great, thanks. Lots of questions coming in. I guess I just suggest folks keep typing away. We'll get to as many as we can. A question about algal and macrophyte respiration. Is it difficult to identify or kind of separate out the difference between algal and macrophyte respiration as oxygen sinks?
So there are some of the time variable models will have separate components for each one of them. The model itself is going to be able to separate it out very easily to like 16 decimal places. The problem is if you were trying to compare it with community respiration as measured in the field, it would be very, very difficult to take the model calculations and try to measure the same thing in the field. You just have to kind of like add them together.
Great. Another question. Since BOD decay rates vary by source, can you include different BOD decay rates from different sources when you're modeling dissolved oxygen?
Yeah, that's a great point, and maybe I didn't make it totally clear. That's why most of the time variable models now and Qual2K have multiple BOD fields, so that you can treat BOD waste streams differently because they will have different properties and decay rates.
Yep. Kind of a more general question. What approach would you recommend for projects, where you perhaps don't have several $100,000 or tens of thousands of dollars to use a complex model, but wish to just kind of get a basic understanding about sources of BOD and whatever, and then its impacts to DO?
Yeah, I mean that really depends upon what the potential outcome is. It really isn't any harder to use a time variable model as it is to use a steady state model. The time variable model uses the same data, it's just there's more of it. You can use any one of these. If you're a first timer and you just want to start looking at the impacts of carbonaceous materials and dissolved oxygen, that EPA DOSEG model is relatively simple to parameterize and just look to see how things behave.
Yep, agreed. So you've throughout the presentation, you've cited a number of references where you've pulled things like reaeration rates and whatnot. Are there new developments in those areas that I guess the public should be aware of, and people doing research in those areas, and how's it being published and reported out?
Yeah, so I'm going to jump up on a soapbox here for a second. You saw from those flux calculations that the primary source of oxygen to the water column is reaeration, and we need more research into one, how to parameterize or describe that phenomena to these water quality models. The most predominant stream reaeration equations are all empirically based, and they're based on research that was done back in the 70s. The USGS has done some more research in that. They have a couple of equations that are applicable to stream riffle areas. But we need more information on doing reaeration in large body systems like lakes and estuaries.
Yep. How does tidal, actually a number of questions on tides and ocean modeling, and I think maybe I'll summarize it as, how would you go about modeling when you have tidal flows or estuary kind of environments where you're getting varying characteristics as the tides come in and out? I guess particularly the question is in reference to SOD as well.
Well, first of all, doing estuaries is going to require you using a hydrodynamic model coupled with a water quality model, because one of the important phenomena that you have to capture when doing water quality there is to make sure that you can handle the density gradients in the system and account for the impacts of stratification on dissolved oxygen. As far as SOD, the SOD model works pretty much the same way. You basically just tell it that you're not in fresh water, you're in salt water, and it uses a different set of equations for calculating it. But it's still driven by the same organic loadings to the sediments.
Yep. And staying on the SOD topic, what do you think is a better measure of SOD? Cores in the lab or chambers in the field? Not sure if you have direct experience with that or not.
I don't have direct experience with that, but I do know that there's a group out of EPA's R&D laboratory down in Gulf Breeze has been working with our field services group that's located in Athens, Georgia, comparing side by side, pulling cores and doing in situ. I believe there's been a couple of papers written, and there's going to be some more. My understanding is that the methodology that you're using for cores now is pretty comparable to the in situ. The nice thing about cores are that you can go out, pull your core, and pull more cores for doing the analysis versus sinking these chambers, because the chambers have to be monitored and babysat by divers, who have to constantly go down and check on them. So I think we're getting somewhere with the cores, and I believe we'll see some more stuff coming out in the literature about that. They're also collecting, as they're doing this, they're also working on collecting some data that's very useful in parameterizing the sediment diagenesis model.
Yeah, Tim, it just has me thinking offhand, do you know of a good resource or website, since there are so many questions on SOD and a number more coming up on monitoring and whatnot, that perhaps we could follow up and point folks towards, or maybe that's a little research we can do on our end?
So the sediment diagenesis model is based upon a book written by Dominic DeToro. I don't know, I think there's like 18 chapters in it. I only understand one. So I don't want to point you there. But there are, I mean, if you search, I think you can find the our field services group's SOP for how you measure it. There are other publications that you can look at about applications of the model. sensitivity analysis with the model.
Great. There's a question specific towards the monitoring for SOD. Do you happen to know kind of the approximate cost for doing a one-day effort to collect and run SOD samples?
It's longer than a day.
Level of effort.
Yeah, it's longer than a day. If you do in situ, I mean, it's going to be when I longer than a day, it's probably going to be about 24 hours by the time that you get the dome down, stabilized, and pull your samples and such. The cost, you know, I used to know what the cost was because a group of people that worked for our field services group opened up their own consulting company, and they were out doing it. But I'm not sure what the cost is for it. It is pretty intensive, though.
It sounds like your group is kind of honed in a method for doing this. Is there someone in your group that folks could contact if needed, or perhaps follow up with you in your email?
Well, yeah, I guess I'm trying to figure out. I don't want somebody to get inundated with questions. If they shoot me an email, I'll hook them up. You know, make sure I get the right people.
Right. Great. All right. Kind of carrying on here. What are your thoughts on the pros and cons of simulating zooplankton as state variables?
Hello, Greg. I tried to call you last week to answer this question. This is one way to get the question answered. You know, we've tried to incorporate mechanistic zooplankton models into our water quality models, and it's been less than fruitful for a couple of different reasons. Zooplankton 1, the critters have the ability to swim. They swim in herds, so it's been difficult. So I don't really try to explicitly model zooplankton. Inherently, the impacts of zooplankton are in the death rates that I calibrate the model to. Or if I'm in a situation where I know I have a seasonal zooplankton population that's in an estuary, I would use time functions to describe what's going on. But as far as doing a zooplankton model, it's been attempted by a couple different groups. I worked with a group in Italy that tried to do one inside WASP, and it didn't work out all that well.
Thanks, Tim. A couple of questions on wetlands here. The first one, is there a model that you recommend over others, perhaps when there are cattails, wetlands, et cetera, along your stream when you're doing your DO modeling?
I'm a little confused. Are we looking at the impacts of attached macrophytes, floating macrophytes, or I mean, cattails are going to, the only real major impact that they're going to have on the water body is actually the water movement, because most of their biomass is above the water. So I'm not quite sure. Maybe
I'd rephrase and say, you know, perhaps it's a little more of a complicated system. Perhaps it's flowing through some wetland areas and such. You know, a little more complicated than a straight run-of-the-river channel.
Yeah, so like the macrophyte model in WASP has the ability to do like floating macrophytes like duckweed and such. So that'll impact oxygen as well as light coming into the water column. It also, you know, the other ones do macrophytes where you have canopy of submerged aquatic vegetation that impact dissolved oxygen at different levels and affect the light through the water column as well. So in that sense, they can do that. We're still not to the point where we have a lot of field data that we can help parameterize and constrain the model, but I'm sure it'll be coming.
Yep. All right. Questions are slowing down, so I think we're kind of getting towards the end here, but we'll do a few more since we have some time. How do you account for seasonal variations from different sources when using a DO model? And perhaps the example they provided is if you have a stormwater discharge that could be very intermittent in nature.
Yeah, so again, you know, the time variable models certainly have the capability of doing that. If you can parameterize the event, the model will, again, take into account the the changes in flow and loadings and predict the changes in dissolved oxygen. So, I mean, that's the reason why we use time variable models, is to take into account episodic events.
Great. A question on basins. Could you maybe give a quick overview on the ability of what's included in what I guess maybe what is basins, and could it be used for doing dissolved oxygen modeling?
So inside basins is, I mean, basins gets confused. Everybody calls it a model, and it's actually a GIS data management system that has a host of different models in it that
Right.
has the ability to help parameterize them. Things like HSPF, which is a watershed model, which does have the capability of doing water quality and dissolved oxygen modeling in riverine systems. It has tools in there for parameterizing a mostly empirical model called aquatox. There's tools in there for building input files for WASP, where it'll build your riverine model network and help populate the flow and load fields. So basins is more of a tool that integrates things together. It has the ability to go out to the web and download information, whether it's land use information or EPA STORET data or USGS NWIS data. So it's kind of like a tool that helps integrate and get things ready for a model.
Great, thanks. A question about a wastewater point source. Their question is specifically a wastewater point source is kind of as creating a DO sag, but the sag is a pretty small area of the stream. And maybe I'll expand it to say, when you've talked about the DO sags over an area of a stream or lake, what's generally the minimum distance that a model could realistically simulate the changes over a distance and time?
So I'm a little. So the length of this, the length or distance or where the sag is expressed is a function of how much of a loading is coming out, how fast does it react, because you may not see a sag until you get 20, 30 miles downstream. So it's a function of what is coming in, how fast does it react, and then what are the hydrodynamic characteristics of the water body. So how far can you model? You can model hundreds of miles relatively easy to investigate where the sag is at.
And I think that's a great answer. I think their specific question is, could a model realistically determine the changes in a sag over a 1 foot area, a 10 foot, 100 foot, 1,000 foot?
So it really doesn't matter what the model is. You're going to break your water body up into pieces. And each one of those pieces is basically an average condition over that distance. So how well you can resolve it is a function of how you break your water body up into those pieces. It's the models that I've talked about aren't what we call near-field models, where they can resolve plumes themselves. They assume everything is completely mixed. So once something comes into one of our segments or cells, it's completely across that. So you do have a little bit of control of how big you make your segments. If you want to either resolve a gradient very, very close, you can make your control volume smaller.
Yep, I think that's what they were asking specifically. Thanks. A few more questions. I think we'll kind of wrap it up in 5 minutes here and just end a little early. So before we get to the last few questions, let me advance a slide. Let me try to do that again. There we go. If you'd like to obtain a certificate of attendance, you can copy down the link shown here. We're going to also email this link out to everyone that's registered for the webinar today. So you'll be getting an email with that link tomorrow. More importantly, you can find future webinar registration links, including the upcoming next webinar on the website that's at the bottom of a page. That's our EPA Water Quality Modeling Workgroup page. A link to that page, like I said, is at the bottom of this slide, and our presentation from today will be archived and stored there soon. And then finally, just a quick flyer for the next webinar, which is going to be on watershed modeling using HSPF. We currently have that scheduled for September 21st, 2016, and Dr. Tony Danigian is going to be presenting that for us. You can get some information on the next webinar at the EPA TMDL modeling website link that you see at the bottom of your page there. The webinar will be at the same time, 1 p.m. Eastern. So we encourage everyone to attend. I believe the next several webinars that we're going to try and do in the series are, as the audience has requested, are going to be more modeling specific focused. In other words, covering how a specific model works and then how you would go about applying that specific model. So stay tuned for more webinars even after the HSPF webinar. And I think I'll just leave it on this slide for a minute and go do another 2 or 3 minutes worth of questions, and we'll end at 3:30. All right, give me 1 second, Tim. Kind of a generic question back to the estuary and tidal flow. So how do tides and tidal flow affect DO and SOD, streams and estuaries?
Well, they're affected in a couple of different ways. Well, the first and most pronounced way is the stratification, the disconnection of the surface waters from the bottom waters caused by fresh water flowing over salt water. That causes the areas, the lower area of water to become less enriched with oxygen or go hypoxic or anoxic. The other issue you have with tides coming in and out is, on an outgoing tide, the water's moving fairly well. On an inbound tide, the water's moving fairly well. But between the tides is where you have slack water, and materials tend to be stagnant or just sitting there, and it has a better chance of pollutants to express themselves on dissolved oxygen and productivity.
Great, thank you. Another question. Several states have been attempting to establish refugia-based criteria for salmonids where the standards couple temperature and DO requirements. The question is, are there models in what you've talked about today that are able to simulate the volume that would fulfill these requirements?
I'm not sure of the volume, but I mean, I know a lot of work has been done out in the Northwest. Greg Pelletier and his group at the Department of Ecology in Washington, they have a special version of Qual2K that will predict the temperature and look at ways to meet the temperature requirements, and I'm certain that they're doing DO at the same time. But yes, there are ways to do that. Temperature is very easy to model, but it's very hard to parameterize if you're not in a wide open flat area. If you're in a canyon area or a canopied area, how do you determine how much light is getting to the stream becomes very, very site specific.
Well, speaking of Greg Pelletier, he's posted another question. What are the pros and cons of simulating zooplankton in estuaries?
Well, I thought I kind of answered that. It's very, very difficult to do. I've did some work on the Neuse River estuary in North Carolina, and they have a pretty healthy zooplankton population there. I couldn't explicitly simulate them as a state variable. I did include them in my model by providing a time function and a grazing rate associated when they were in the area, when they were typically in the area. But typically, the effects of zooplankton are kind of calibrated into the death rate, like I said previous.
And I think we'll do one more question and just call it quits a little early. we've talked in previous webinars about, how to calibrate and how to determine when you have satisfactory results, and are there criteria for those things. So could you comment maybe more specifically on when modeling DO? How do you know when you've got a good enough calibration? Are you comparing your calibration to some kind of rates or kind of predefined expectations, or maybe just expand on what we've talked about before, but with a DO bent?
Yeah, and maybe at some point in the future we can have a webinar on talking about calibration and such. But how do I know when it's good enough? It's either when I ran out of time or I ran out of money. I mean, that's the kind of the way it is. It's a bad idea to go into any modeling project with a preconceived idea of what is going to be good enough, because good enough is a function of how much information you have about the system, but also how much information you have to model the system. That's both hydrologically, hydrodynamically, water quality, and as well as how much knowledge you the person doing it, has. I mean, I've seen water quality studies where the model was being asked to resolve a question that you couldn't resolve with a DO probe. So that's probably a bad idea. So for DO in a riverine system, you probably should be able to in a steady state model, you should be able to get it fairly close. In a dynamic time variable model, maybe not so close because of so many things changing. But I can't give you a number. I can't say, listen, if you're within 1 milligram per liter, you're good. That's not how it works. It's when you apply models to answer questions, you're going to be engaging stakeholders, and you've got to be able to convince them that the model is representing the system the best it can. Or if you run out of time and money.
And I think I would add, we talk about this a bit in the first 3 webinars, which again are archived and posted to that website that you see on your screen now. So I'd highly recommend folks to go back, and if you have questions kind of about calibration in general and setting performance standards, to go back and have a listen to those, and hopefully those would help answer your questions. And if not, yeah, maybe if we get some feedback from folks, maybe a calibration webinar would be a good thing to do sometime in the future. So with that, I would like to give Tim, or say to Tim, a great big thank you. We really appreciate you doing this today. This concludes our webinar. Thank you to everyone who joined us, and hopefully we'll have you back on one of our upcoming webinars, particularly on the one on the 21st. So thank you, everyone, and goodbye.